Greetings and welcome to another lecture in Introductory Psychology. This one involves vision, specifically color vision. Now, researchers have been trying for a long time to figure out exactly what's going on biologically, physiologically, when it comes to us being able to perceive colors. And one of the oldest theories of color vision is called the Trichromatic Theory. And like a lot of theories in psychology, it's named exactly what it is. Tri meaning three, chroma meaning color. Now, the Trichromatic Theory says that we use three different types of cones to see colors. Now, the three different types of cones we have are red cones, green cones, or blue cones. Or actually, they should be called cones that are sensitive to red wavelengths of light, or wavelengths of light that correspond to us seeing red. It gets complicated. Cones that are sensitive to green light, cones that are sensitive to blue light. Now, the idea is that with these three colors, when they are mixed and matched, we can get every other color in the color spectrum. Now, this may sound familiar if you've taken an art class. You may have heard about what are called primary colors, where if you take those primary colors in paint, for instance, you can mix them together in certain amounts and make every other color. And the principle is exactly the same, except if you remember those primary colors, they were not red, green, and blue. They were red, yellow, and blue. And the reason for that is that paint mixes together very differently than light. If you take red and yellow and blue paint and you mix them all together, you're going to wind up with a dark color like a dark brown or a black sort of color. If you take red and green and blue light and mix them all together, you come up with white. They mix together differently. And it turns out that we do indeed have three different types of cones. We have cone sensitive to red, cone sensitive to green, cone sensitive to blue, and also that it does look like this is how we see color. Except things in science are never that simple, or at least for the most part, they're not that simple. This one certainly isn't, because that explains an awful lot, and it does make sense when we look at our retina and our cones and such, but it doesn't explain one thing, and that's a very interesting phenomena called afterimages. Now, an afterimage is not like, when someone takes a flash picture and you don't see anything for a while. That, by the way, is caused by the fact that your cones have been overstimulated and they're trying desperately to recover. But an afterimage is, I actually have an example here because it's really kind of cool. And it's nice that I can do this on a video. What I would like you to do is I would like you to stare at that white dot in the middle of that flag. No, nothing's going to jump out at you. Nothing's going to scream at you. There's going to be no soundtrack edition here. Okay? This is a different kind of YouTube video. But I would like you to stare right at that white dot for just a little while. Keep staring. I'm still here. And just keep staring at that dot. Just keep staring. Now, this should really go on for like 30 seconds, but I really don't want to put 30 seconds of dead air here. So if you want to do it correctly, you should pause this and then, count to 30 all the while staring at this flag and then go back to it. But keep staring. Isn't that cool? If you possess normal color vision, and there's no guarantee that you do, you should have seen something very interesting when that flag went away. Let's do it again. Stare at the white dot. Keep staring. Like I said, you might want to pause it, particularly if you didn't see it before. Pause it and stare at it for like a minute, and then start it back on up. What you probably saw there was an afterimage. What you saw was an image of the American flag, but instead of being green, yellow, and black, it was red, white, and blue. You can't explain that with the Trichromatic Theory. So researchers came up with another one. They call it the opponent process theory. Now, the opponent process theory suggests that we see colors through opposites, essentially, through opponents in a way. That we take these primary colors, and they actually added yellow in, by the way, because it does appear there are green cones that are a little more sensitive to the yellow end of the spectrum. But you take these colors, and these colors are in opposite pairs. So a blue cone is paired with a yellow cone that's pretty much right next to it. And remember, when we're talking microscopic here in terms of cones, you have millions of cones in each retina. So you could have a blue cone right next to a yellow cone that are wired together, and a red cone right next to a green cone that are kind of wired together, in the brain anyway. Because when you look at those colors, you may notice that those are what are called complementary colors. That basically those are opposites. If you have a color wheel, if you had art class, you may have looked at this. You'll notice that red and green are at opposite ends of the color wheel. And actually it's purple and yellow that are on opposite ends, but we got to go with blue because those are the cones that we have. You got to work with what you have. And the way this essentially works is that when you have these complementary colors, they don't really get mixed together a whole lot. You know, we don't necessarily say something is kind of yellowish blue. We may say it's green, but we don't say it's yellowish blue. In the same way, we don't say something is kind of reddish green. We may say it's kind of brown, but we don't say it's reddish green or greenish red. That's just kind of weird. And so these opposite pairs are wired together in the brain, in the occipital lobe of the brain, in the visual cortex. Now the idea is, since these colors are opposite, you can only really be looking at one at a time. You can't have both red and green light be on the exact same microscopic spot on your retina. Now, if you're going to ask about edges, and what about some plaids you've seen, and what if you put a line of red and a line of green right next to one another, that's a little bit more complicated than I can get to in this video. Look into maybe not an anatomy, but more of a physiology or a sensation book, and then you could learn about all of the ways that we see edges and colors right next to each other. I will tell you, though, that when you're talking microscopic, even when green and red are right next to one another, there's still a line. There's still, the odds are you're not going to have red and green fall on the same pair. So if the red cone or the blue cone is being stimulated, then the yellow one, we'll stick with blue and yellow, since red and green you get into color weakness. When the blue cone is being stimulated, then the yellow one isn't. So the blue cone may become more active in sending signals, and the yellow cone become less active. Or if you're looking at yellow, the yellow cone becomes more active, the blue cone becomes less active. It's almost like a seesaw. If you've ever played with a seesaw, or what we used to call them teeter-totters, or a scale with balance, where you put weight on one side. If you want to balance it, you have to put weight on the other. Well, if you put more weight on one side, that side goes down, the other side goes up. But if you take that weight away, maybe put weight on the other side, then the reverse happens. So when one is activated, the other is suppressed. Now that would give us after images, because after a while, if you've been looking at blue and your blue cones are going blah, 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 they've just been sending blue signals like crazy, it's possible that when you look away, there could be a little bit of a bounce. They're not going to go immediately back to neutral, in the same way that if you have one of those scales, or you have a seesaw or teeter-totter, if somebody is sitting on one end, even if there isn't anybody on the other end, okay, and you get up very quickly from that one end, it's not going to just immediately go back to level. It's going to wangle back and forth a little bit before it balances out. And that rebound, that wangling back and forth before they balance out, can give you the after image. For just that little bit, for just that little flash, you see that flag hanging in front of you on the white space. Because think about it. Let's go back to that slide real fast. One more time, let's stare at this slide. I know it's a little boring, but it's also really kind of cool. Stare at the slide. Note that yellow became blue, that green became red, and that black became white. Those are the opposite pairs. And if you don't trust me, try it again. But we're not going to go through that again because we've already been. So opponent process basically is we have opposite cones wired together in the brain. Trichromatic is that we have three types of cones that respond differently to different wavelengths of light. Well, the big question whenever we have these kind of theories, we have more than one theory for a particular process, is, well, which of these is correct? In science and in psychology, you will often find that the general answer is both of them. And that is indeed true here. Because what seems to be going on here is Both of them are working, but in different places. The trichromatic theory seems to be working at the level of the cones in the retina, because we do have three types of cones. And they are sensitive to different wavelengths of light. And by the way, if you haven't listened to my visual problems lecture, red-green color weakness, which a lot of people have, is actually a defect in the cones. The brain is fine, but the cones don't work properly, which causes people with red-green color weakness to basically see the same color for red and green. It looks the same to them. So trichromatic works at the level of the cones. But those red cones and those green cones and those kind of yellow-green cones and those blue cones are wired together in the brain in opposite pairs. So both of these allow us to see color.